eye development is fundamentally about chemical and physical signaling between adjacent embryonic tissues.
So one group of cells says something to its neighbors.
Exactly. One tissue signals, the adjacent tissue responds by changing its fate, differentiating or migrating, and then that tissue signals back or signals to a new neighbor, initiating the next stage of development. It's a dynamic domino effect sequence where proximity and timing are, well, they're everything.
It's a beautiful dance, but one that requires 4 specific types of dancers, right? The sources identify 4 key tissue contributors right out of the gate, establishing the foundation for everything we're about to discuss.
That's right. And it's crucial to get these players straight from the beginning because they all have very distinct roles.
Okay, so player number 1.
The first and perhaps most critical player is the neuroectoderm of the forebrain. This is the originating source material for all the sensory components. The neural retina, which receives and processes light, and the critical pigmented structures that absorb stray light. It's the brain, essentially, reaching out to see the world.
Okay, brain tissue forming the sensor. Then you have the tissue right on the surface.
Right. Player number 2 is the surface ectoderm. This tissue, lying directly over the developing eye field, is what responds to the brain's signal. It's responsible for forming the transparent components that focus the light, specifically the lens and the outermost layer of the eye, the corneal epithelium.
So the neuroectoderm is the film, and the surface ectoderm is the lens.
A perfect analogy. Then third, we introduce the supporting cast, and these are highly versatile migratory cells. The intervening neural crest mesenchyme. These cells are not just structural filler, they are the architects of the connective tissue. They form the fibrous coats like the sclera, the bulk of the cornea, and the stroma of the iris. Their ability to migrate is just key to building the complex anterior segment.
And finally, number 4.
The intervening mesenchyme, which also contributes heavily to those fibrous coats, specifically providing the structural support and blood vessel components, particularly in the posterior parts of the globe.
So to build an eye, you need the neuroectoderm for sight, the surface ectoderm for focus, and the neural crest and mesenchyme for protection and support. A 4-part harmony.
And the complexity starts right there. The fate of the neuroectoderm depends on the surface ectoderm, and the integrity of the lens, which is from the surface ectoderm, depends on the surrounding neural crest cells. This constant induction is what we will track.
All right, let's rewind then to the very earliest stages, before the eye even looks like a cup or a sphere when it's just a potential. The sources start by discussing the genetic drivers that establish what's called the eye field.
Right, and it's not a random spot. It's a broad anterior domain of neuroectoderm that is specifically characterized by turning on a very particular set of transcription regulators.
These are the master switches.
The absolute master switches. We are talking about genes like PAPIC-XDIX, 6IX33s, and ORX22s. These genes are fundamentally responsible for establishing the potential to form the optic vesicles later on. Their job isn't to start the folding process yet. Their initial role is much more fundamental. It's about establishing the correct anterior identity.
And that's where the crucial subtlety comes in. They don't just say, make an eye here. They're also involved in an internal debate about where this tissue should be.
Exactly. That's a great way to put it. These regulators are vital because they are involved in actively repressing competing signals, specifically what embryologists call caudalizing WNT signaling.
Okay, break that down. Caudalizing.
Think of the embryo's body plan. You have a head end, or anterior, and a tail end, the caudal end. WNT is a family of signaling molecules that typically promote caudal or trunk-like identity. It's the signal that says, make spinal cord, make posterior structures.
So it's the make something tailward signal.
Precisely.
Yeah.
If pan-size 66 and sexing 83633 didn't repress these signals in the very front of the neural plate, that tissue would lose its ability to form forebrain structures, including the eyes. It would get confused and try to become something else. So these genes essentially lock in the head identity.
That context makes it much clearer why those transcription factors are so critical. It's a fight for anatomical location. But even once the forebrain identity is established, the eye field starts as a single structure, right?
Yes, a single continuous field across the midline. So the next challenge detailed in the source material is carving that single potential eye field into 2 bilateral domains. This requires a very famous secreted protein.
And that would be?
That is the incredibly powerful signaling molecule, sonic hedgehog, or SHHL. SHHL is secreted from the precordial plate structure located right along the midline of the embryo. Its function here is to pattern the prosaiciocephalon, the front of the brain, and crucially to instruct that initial single neuroectodermal field to split down the middle, separating the future left eye from the future right eye.
So it's like a molecular sculptor carving out two distinct zones from one block of marble.
That's a perfect analogy. It sends out a signal from the center that essentially says don't form eye tissue here, pushing the eye-forming potential out to the sides.
So if synergic is the molecular divider, what does it mean for the listener when this process fails? What happens if that signal is too weak or just not there?
The consequence of losing adequate synergic activity is catastrophic for midline separation. The sources clearly link the failure of this genetic instruction to severe cranial anomalies, which is a powerful clinical connection for the learner. The most severe manifestation is cyclopia.
Cyclopia, a single central eye.
Exactly. The single eye field fails to separate, resulting in the formation of only a single central eye and fusion of the associated orbital structures. It's a direct physical result of that midline signal failing.
And it often goes hand in hand with an even broader problem, doesn't it?
Yes, cyclopia is usually part of the spectrum of holoprosencephaly, which is the incomplete separation of the prosencephalon, the forebrain itself. Since MIH is required to pattern the entire frontal midline, its failure means the entire front of the brain remains fused or improperly divided. That's why the visual system defect is such a striking marker for this broad developmental failure. It just underscores how early and how tightly controlled this genetic patterning must be.
Given how easily things can go wrong if these signals are lost, it raises an important question. How robust is the intrinsic programming of these cells? The sources provide a really striking discovery from lab settings that demonstrates the inherent organizational power of these early cells in a way that's quite shocking.
This is a truly profound aha moment about cell memory and potential. Researchers took in vitro embryonic stem cells, cells that haven't been told to become anything specific yet, and they supplemented them with the correct growth factors and a structural support matrix, something like matrigel.
So they just gave them the right chemical soup and a scaffold to grow on.
That's it. And what the sources note is that these cells spontaneously underwent dynamic shape changes, essentially self-organizing to form a two-layered optic cup structure.
Wait, hold on. If this entire deep dive is about inductive interactions, one tissue signaling another, how can the neuroectoderm just spontaneously start making an optic cup in vitro without the surface ectoderm there to tell it to start?
That is the core takeaway. It's a fantastic question. It suggests that the program for forming the cup, the folding, and the initial stratification is deeply encoded within the neuroectoderm itself, provided the environmental factors, the growth factors we supplied, mimic the early necessary conditions.
So the blueprint is already inside the neuroectoderm cells.
It seems so. The resulting tissue is phenomenal. It mimics postnatal retina, forming a stratified architecture with appropriate neural layers and even primitive synapses. It's an eye in a dish, built from its own instructions.
So the surface ectoderm might act more as a trigger for the lens specifically, but the underlying neuroectoderm already carries the architectural blueprint for the cup and retina. That's incredible.
Exactly. And the sources confirm similar results have been achieved using human embryonic stem cells, producing not just a layered retina architecture, but also fully functional retinal pigment epithelial cells. This is crucial for regenerative medicine because it suggests the inherent capacity to build a functional retina is incredibly powerful, locked within the identity established by those early transcription factors like PX60LRs. This capacity for self-organization is far more sophisticated than we might expect from simple cell aggregates.
Having established the importance of that genetic programming, let's now look at the physical mechanics this allows. The actual morphing of the neural tissue into the eye structure. The sources mark the earliest morphological sign of eye development very precisely.
Yes, the very first observable physical step is the thickening of the diencephalic neural folds at stage 10. This happens around 28-29 post-fertilization days, when the embryo has about 7 to 8 somites. This thickened area is the optic primordium, or eye field, extending across both sides of the neural plate.
So just a thickening at first.
Just a thickening. Then a slight transverse indentation, the optic sulcus, appears on the inner surface of this primordium, signaling the very beginning of the outward push.
So it starts as a gentle indent, but quickly becomes a massive outward projection.
A huge projection. By stage 13, so just a few days later, around days 31-33, the walls of the forebrain, specifically a segment embryologists call neuromere diencephalon 1, begin to balloon out or evaginate laterally. This culminates in the formation of the full optic vesicles.
And these are projecting toward the surface ectoderm.
Directly toward it. And crucially, the lumen, the hollow inside of each optic vesicle, remains continuous with the cavity of the diencephalon, the developing brain. It's still physically connected.
And this evagination is the critical first step in that inductive conversation we talked about. Once the optic vesicle touches the surface ectoderm, the signal is passed, the lens begins to form, and then the vesicle itself undergoes this dramatic process of invagination folding in on itself to create the optic cup.
The mechanics of this folding are remarkable. A great way to visualize it is to imagine pushing your thumb into a hollow, slightly deflated rubber ball. The outer layer becomes the convex surface, and the inner layer where your thumb is becomes the concave receiving surface.
And this internal folding leads to what you called the layer flip. Let's elaborate on that, because it's easy to miss how strange it really is.
It is deeply strange, and it defines the architecture of our vision. In most epithelial structures, the apical surface, the top surface, faces the outside world or a lumen. The basal surface rests on a basement membrane. But when the optic vesicle folds, the formerly apical or luminal surfaces of the two layers, the presumptive neural retina and the retinal pigmented epithelium, now end up facing each other.
So the two top surfaces are now touching in the middle.
Exactly, separated by a greatly reduced internal cavity called the intraretinal space. This means the photosensitive ends of the eventual rods and cones will be pointing away from the light source embedded against the pigmented layer at the back.
That is a fundamental constraint of vertebrate vision. The light has to travel through all the neural layers before it hits the photoreceptors. It seems completely backward.
It's an evolutionary artifact of this folding process. The outer layer, influenced by signals from the surrounding mesenchyme, becomes the highly specialized retinal pigmented epithelium, or RPE, and the inner layer, facing the lens, becomes the thick, light-sensing neural retina. And the sources highlight the functional consequence of that layer flip, which is the junction between the sensory retina and the pigmented epithelium is less firmly attached than the attachment of the pigmented layer to the underlying mesenchyme. This vulnerability is why this specific junction is the site of pathological retinal detachment in the adult eye. It's an architectural weakness we inherit directly from this folding process.
Wow. Okay, so now we address the structure of the cup itself. It isn't a perfectly closed sphere initially. There's a crucial gap.
That's right. The cup remains incomplete, connected to the brain via the optic stalk. And the ventral aspect of the cup, so the underside, opens into a wide groove known as the optic fissure, or choroid fissure. This seems counterintuitive. Why leave a wide open groove in the bottom of your protective structure?
Exactly. Why is this fissure necessary? It seems like an intentional breach in the structure that you'd want to avoid.
It is absolutely intentional and completely essential. The optic fissure is the critical vascular gateway. We can go back to that construction analogy.
Construction entrance.
Precisely. This wide groove serves two primary purposes. First, it allows surrounding mesenchyme to migrate inward to contribute to the vitreous humor and the surrounding fibrous tissue. And second, most importantly, it permits the ingressing hyaloid artery to enter the developing globe and nourish the rapidly growing interior structures, especially the lens. The entire formation of the cup involves differential growth to wrap around this vascular ingress point.
So the fissure is temporary, a necessary evil for vascularization. Once the hyaloid artery is in place, the fissure's job is done, and it has to close.
It must close. And failure of this fusion is what leads to one of the most recognizable and potentially debilitating congenital anomalies, congenital coloboma. This condition, meaning a deficiency, results when the fissure margins fail to meet and fuse.
And how does that fusion happen?
It's a process characterized by apoptosis-programmed cell death at the edges of the fissure. The cells that form a temporary bridge have to be eliminated for the two sides to fuse seamlessly.
So if that apoptosis fails, or the fusion just doesn't happen, what are the physical manifestations of coloboma?
It results in a gap or a notch, typically inferiorly, in corresponding structures like the choroid, the retina, and often the iris. This can give the pupil a characteristic keyhole appearance. Functionally, if the retina itself is incomplete, the person will have a corresponding blind spot, a visual field defect.
And it's often associated with the eye just being too small overall.
Exactly. It's often associated with microphthalmia, the condition of having abnormally small eyes, which indicates a general failure of growth signaling.
That microphthalmia link brings us back to the genetics. The sources mention specific genes linked to the sheer size of the developing eye.
Yes, the sources highlight that reduced optic cup growth, which often underlies microphthalmia, can be traced back to mutations in the homeobox gene VSX282-OPA. This gene is absolutely crucial for the proliferation and growth of the neural retina. If the retina doesn't grow enough, the cup simply cannot expand properly, leading to small eyes.
So it's a growth factor problem at its core.
It is. They also link another gene, SOS2E2-opiate, to the whole spectrum of anophthalmia, which is absence of the eye, microphthalmia, and coloboma. This clearly demonstrates how the initial genetic controls over cell proliferation dictate the entire final physical size and integrity of the visual organ. It's a powerful illustration.
Now that we have the cup formed, the real specialization, the molecular war for cellular identity, begins. We have two layers that must differentiate into radically different functional components, the sensory retina and the pigmented epithelium.
Let's break down the two fates, starting with the retinal pigmented epithelium, RPE, the outer layer. Its identity has to be established early and definitively. It's destined to remain a thin, single sheet of cells, and its function is to support the photoreceptors and absorb stray light, preventing it from bouncing around inside the eye.
And there's a specific gene driving this fate.
Yes, the critical molecular driver for this pigmented fate is the MITF-YID gene. This gene steers the cells toward pigmentation, and we can actually see the result structurally. These cells acquire their pigmented melanosomes at stage 17.
Okay, so that's the outer layer. Meanwhile, the inner layer, the neural retina, is taking the complete opposite path. It's undergoing massive, rapid proliferation.
A huge amount of proliferation, forming a thick, pseudostratified neuroepithelium. When we say pseudostratified, we mean that although the nuclei are at different levels, every single cell extends from the apical surface right down to the basal surface. It's a very organized crowd.
And the molecular driver here is different. It's an antagonist to gamma-TFA.
It is. The molecular driver here, turning the cells into the light-sensing structure, is the VSX2A2 gene. The proper patterning of the optic cup relies entirely on this regional molecular antagonism. MANIT-F5 active in the outer layer, VSX2A2FX active in the inner layer, setting the two distinct fates.
The neural retina then has to organize itself into this complex layered structure to handle light processing. The sources describe the initial internal organization.
Initially, it has two primary zones. First, you have the outer nuclear zone, which is where the dividing neuroepithelial retinal progenitor cells reside. This is the germinal zone, the factory. Second, you have the inner marginal zone, which is initially empty, devoid of nuclei.
A cell-free zone.
For a time. But by stage 18, cells start to exit the cell cycle in the outer zone and begin to migrate, invading that marginal zone. This process results in the formation of the distinct inner and outer neuroblastic layers, the beginning of stratification.
What's truly remarkable here, and what speaks to the intense organization required for vision, is the cell lineage. The sources emphasize that all 7 retinal cell types, that's every photoreceptor, interneuron, and glial cell in your retina, derive from a single, common, multipotential progenitor cell.
One parent cell for everything. And their development is not a free-for-all. It follows a highly conserved, specific birth sequence across species. This is a critical piece of information for the learner. The retina is built in distinct phases.
So who gets born first?
The early-born cells are the foundation. These are the ganglion cells whose axons will bundle together to form the optic nerve. Then you get the amacrine cells, the cone photoreceptors for high-resolution color vision, and the horizontal cells. These are the initial processing layers.
And the ones born later tend to be the higher volume or supporting cells, I imagine?
That's a great way to think about it. The later born cells are the bipolar cells, which transmit signals from the photoreceptors to the ganglion cells. Then you have the rod photoreceptors, the vast majority of photoreceptors, crucial for low-light vision. And finally, the vital support structure, the Miller glial cells. This timing is critical. If a gene mutation messes with this schedule, the entire laminar structure can be compromised.
And the process of establishing the layers then involves these newly born cells migrating from their birthplace at the apical surface to their appropriate final layer.
Yes, and the layers separate sequentially. The ganglion cell layer separates first, establishing the inner plexiform layer where synapses happen. Later, the inner nuclear layer, which contains the amacrine, horizontal, bipolar, and Miller glial cells, separates from the outer nuclear layer, which houses the rod and cone photoreceptor nuclei. This creates the outer plexiform layer. It's this incredibly precise, organized migration that ensures the wiring is correct.
Now for the final stage of specialization, photoreceptor maturation. When are we actually ready to see?
That's the key question. Mature retinal neurons appear first in the central part of the retina, the future foveal area. All the named layers are generally identifiable by postmenstrual weeks 29-32. However, and this is a key developmental constraint the sources emphasize, photoreceptor cells continue to differentiate and mature structurally after birth.
So a newborn's retina is still a work in progress?
Very much so. This prolonged development, particularly the lengthening of the outer segments of the rods and cones and the migration of cells to form the foveal pit, is what leads to the gradual, dramatic improvement of visual resolution and sensitivity over the first 1 to 3 years of life. A baby is literally building a clearer picture of the world day by day.
Here's where we circle back to that aha moment that bridges embryology and future medical application, the plasticity secret. We detailed how the RPE and neural retina layers committed to distinct fates driven by 1ITF1 and VSX2 along. But the source material makes a crucial observation about the non-finality of that commitment.
It's one of the most exciting findings. Despite that very early and very firm molecular commitment, the RPE, the pigmented non-neural outer layer, retains the potential to become neural retina if the embryonic retina is wounded.
So it remembers its other possible life path.
It does. This is a powerful conserved mechanism observed in lower vertebrates like salamanders, which can regenerate their retinas. In mammals, it's much more limited, but the potential is still there in the embryonic state.
That is fascinating. But if the potential is still there, why can't we just wound an adult retina and watch it regenerate new neurons? What is fundamentally different about the environment or the cell state in the adult?
That is the essential billion-dollar question that drives regenerative research. What the sources show is that the commitment, while firm, is not terminal in the embryonic state. As the RPE matures and differentiates further, the latent neural potential appears to be actively repressed by the surrounding adult environment and inhibitory signals from neighboring cells.
So the system actively shuts down that potential.
Yes, but this plasticity secret gives us a molecular roadmap. If we can understand and reactivate those early VSXT2 switches while repressing MITAFO in adult RPE cells, it might unlock the capacity for self-repair years down the line. It proves the genetic program isn't deleted. It's just silenced.
The retina is useless without a transparent structure to focus the light onto it. So let's talk about the lens. Its development is the classic example of that powerful inductive interaction we started with.
It's the textbook case. The optic vesicle signals to the adjacent surface ectoderm. That signal exchange is rapid and effective. The overlying surface ectoderm thickens into what we call the lens placode.
Like a little circular patch.
Exactly. Then the placode invaginates, forming a pit, which eventually pinches off entirely from the surface ectoderm to form the lens vesicle, which is a hollow ball of actively proliferating epithelial cells.
And once that vesicle pinches off, the surface ectoderm just reforms right over it, creating the future corneal epithelium. It's an almost surgical-like detachment, leaving the placode material suspended inside.
It's beautiful. Then comes the unique transformation to create transparency. This involves the formation of the primary lens fibers. The cells of the posterior wall of that hollow lens vesicle dramatically thicken, they lengthen, and they push forward until they fill the entire vesicle cavity. This happens around stage 18.
And this is truly the aha moment of specialized cell biology. These cells, unlike nearly any other cell in the body, sacrifice their standard cellular machinery to achieve a singular functional goal: transparency.
They undergo a unique terminal differentiation. They lose their mitochondria and, most remarkably, their nuclei entirely.
They just get rid of their own nuclei.
They eject them. They become metabolically quiescent and are densely packed with extremely high concentrations of specialized soluble proteins called crystallins. The precise ordered packing of these crystallins is what eliminates light scattering and renders the lens transparent. By stage 22, this process is largely complete, reducing the original vesicle cavity to a mere slit.
So those primary fibers form the non-dividing, non-metabolic core of the lens. But the lens isn't static. We know it keeps thickening throughout our lives.
That continuous growth is driven by secondary fiber growth. Cells in the equatorial region that ring around the middle of the lens continue to divide. These new cells elongate and are layered onto the existing primary fibers, like layers of an onion. This proliferative process is sustained by the continuous activity of cells in the anterior epithelium and, crucially, continues throughout adult life.
And this ongoing process is why, decades later, the lens is much thicker and less flexible, which is implicated in age-related vision changes like presbyopia.
Absolutely. The sources detail the complex regulatory process guiding this growth. Lens polarity and sustained growth are tightly regulated, dependent on the differential distribution of soluble factors like BMP, retinoic acid, and N-H-Ehlers in the aqueous humor in front and the vitreous humor behind. This differential signaling guides the specific direction of elongation, ensuring the lens maintains its correct shape.
And when this complex genetic and structural process fails, The result is pathology we know well.
Yes, failure in development often results in congenital cataracts clouding of the lens. The sources connect this pathology strongly with mutations in genes encoding transcriptional regulators like FOXE33, which is critical for anterior lens formation, and most commonly, mutations in the crystalline proteins themselves. If the crystallines aren't formed perfectly, they can clump together, scatter light, and create opacity.
Now we address the life support system. The rapidly developing, energy-hungry retina needs blood supply, and the source material describes a complex evolution involving two distinct, interwoven vascular systems, one of which has to be built, used, and then deliberately destroyed.
Right, the temporary system. This is supplied by the hyaloid artery. As we previously discussed, this artery enters the eye through that essential breach, the optic fissure. It transiently supplies the developing lens capsule and the inner part of the retina during that crucial embryonic period. It is the necessary lifeline early on.
But the adult lens is avascular for transparency. That means this entire system has to be dismantled, which seems incredibly risky. What if it doesn't go away properly?
It's a great question, and sometimes remnants do persist, but it's a remarkable and necessary act of self-destruction. The hyaloid artery eventually regresses entirely via apoptosis-programmed cell death. This active macrophage-dependent tissue remodeling process occurs around postmenstrual weeks 29-32. Once this vascular capsule surrounding the lens disappears, the lens becomes entirely reliant on diffusion from the aqueous and vitreous humors for nutrition. This regression is non-negotiable for achieving optical clarity.
But the entire artery doesn't disappear, right? Some part of it sticks around.
No, a small proximal part of the hyaloid artery persists in the adult as the central artery of the retina. It runs through what's called the hyaloid canal, a transparent, fluid-filled remnant that passes through the vitreous. But the extensive network that once surrounded the lens is completely gone.
So what replaces the temporary supply? Let's talk about the two permanent supply systems that sustain the retina.
First to develop is the choroidal vasculature. This develops earliest, localizing around the retinal pigmented epithelium around stage 18, originating from the surrounding mesenchyme. It forms a complete, dense vascular plexus relatively early, by postmenstrual weeks 21-22. This system is crucial because it supplies the outermost, most metabolically demanding layer of the retina, the photoreceptor cells.
And second, the system that spreads across the inner surface of the retina.
The retinal vasculature. This forms later, starting around postmenstrual week 15, and the sources detail two different ways this network expands, which is quite interesting.
How so?
Well, the initial inner plexus of the retina forms via vasculogenesis. That's the creation of vessels de novo, from scratch, by the aggregation of spindle-shaped vascular mesenchymal cells that emanate from the optic disc.
So they build the central hub first.
Exactly. Then, once this initial central network is established, the temporal and peripheral expansion relies on angiogenesis. This is the sprouting of new vessel segments by endothelial cell migration from pre-existing vessels, growing tangentially through the inner neuroepithelium towards the periphery.
So vasculogenesis builds the trunk and main branches, and angiogenesis grows all the little twigs out to the edges.
A perfect way to put it. This process is complex and, as we'll discuss later, highly vulnerable to disruption, particularly in premature infants.
And here is a crucial functional detail. Despite this dense network covering the retina, the sources note a definitive, necessary exception, the foveal area.
That exception is absolutely essential for high-resolution vision. The foveal area, which is responsible for our sharpest visual focus, never develops retinal vasculature. It's in a vascular zone.
Why?
To maximize light transmission. This design ensures that light heading for our most sensitive cones isn't interfered with, scattered, or absorbed by red blood cells in overlying vessels. It's a final, critical piece of engineering for optical perfection.
We've thoroughly covered the back of the eye and the middle. Now we move to the front, the anterior segment, which includes the cornea, iris, ciliary body, and the intricate drainage system for aqueous humor. This structure is defined by a massive migratory event.
Absolutely. The formation of the anterior segment is largely dictated by the extensive migration of neural crest cells. These are the great travelers of the embryo. They migrate ventrally, surrounding the optic cup and positioning themselves between the surface ectoderm and the lens. These neural crest cells contribute to the majority of structures here: the ciliary body, the iris stroma, the crucial cornea stroma, and the iridocorneal angle tissues.
Let's focus on the cornea, which is a masterpiece of transparency built from 3 distinct parts, each with a different origin. It's a great example of that whole process.
It is. The cornea has a trilayer origin. The outermost layer, the epithelium, is derived from the surface ectoderm, the same tissue that formed the lens plaque. The bulk of the cornea, the stroma, and the innermost layer, the endothelium, differentiate from that invading neural crest mesenchyme.
And the stroma is the real key to transparency. How does a sheet of tissue achieve that? It's not just about being thin.
No, it's about hyperorganization. The stroma develops a remarkably regular array of collagen fibers, the lamellae, which are specifically oriented to minimize light scattering. But the most critical component for maintaining transparency throughout life is the corneal endothelium.
What does the endothelium do that's so important?
It's an organized single monolayer of cells that acts as a pump. Its entire job is to maintain transparency by constantly regulating the water content of the stroma. The cornea must be kept at a precise state of dehydration. If that endothelial layer is compromised, the pump fails, the cornea swells with excess water edema, and it instantly becomes opaque. Vision depends on the perfect function of this single-cell layer.
Amazing. Next, the ciliary body and the iris. These are compound structures that control light entry and produce fluid.
And again, they showcase dual origins. Their epithelial components are derived from the peripheral rim of the optic cup. But the surrounding connective and supporting tissue, the mesenchyme, which is mainly neural crest, forms the vascularized folds of the ciliary body and the stroma of the iris. The ciliary body is also the anchor point for the fibers that hold the lens in place.
And the ciliary body's primary active function is the secretion of aqueous humor, the fluid that fills the front of the eye. This leads us to the incredibly precise and critical system for regulating intraocular pressure.
Aqueous humor circulation is a closed system that has to be in constant balance. It flows from its production site in the ciliary body through the pupil and into the anterior chamber. If it is produced, it must be drained. This occurs mainly via the iridocorneal angle, which is the intersection of the iris and the cornea.
And what's the mechanism for drainage? It can't just soak away.
No, the fluid is filtered through a specialized porous structure within that angle called the trabecular meshwork. This meshwork is a delicate filter composed of trabecular beams formed entirely by those migratory neural crest cells. The fluid filters through this meshwork and is ultimately drained into a collector channel, the scleral venous sinus, often known as the canal of Schlemm.
So if the neural crest cells don't form that trabecular meshwork or the angle correctly, if the architecture is just slightly off, the fluid can't drain, what are the clinical consequences?
Precisely. The outflow dynamics are entirely dependent on the perfection of this neural crest-derived drainage system. The sources link genes like FOFCAPSOID and PITX282, which regulate neural crest cell migration in this region, to developmental angle malformation. This anatomical failure often leads to secondary glaucoma, a persistent, damaging rise in intraocular pressure, as well as issues with the iris and cornea.
One final fascinating detail about the iris that connects back to the neural crest cells: color. The sources note that iris coloration is a postnatal event.
It is, and it's an interesting physical change that you can observe in infants. The final iris color develops after birth and is dependent on two factors: the pigment from the posterior epithelial layer, which is fixed, and the migration and maturation of neural crest-derived melanocytes into the iris stroma.
So what determines blue versus brown?
If only the deep epithelial pigment is present, the eye appears blue due to light scattering in the stroma. It's the same reason the sky is blue. If the neural crest-derived melanocytes migrate fully and produce additional melanin pigment in the stroma on top, the eye appears green, hazel, or brown. The depth of color is literally the depth of melanocyte activity.
We've covered the globe. Now we zoom out to look at the incredibly complex supporting architecture: the eyelids, the tear ducts, and the specialized extraocular muscles that allow us to track and focus. Let's start with the eyelids.
Eyelids are formed from two cutaneous folds of surface ectoderm, with a core derived from neural crest mesenchyme. Their initial function isn't protection from the outside world, it's consolidation. They come together and fuse over the cornea around postmenstrual weeks 10-11, enclosing what will become the conjunctival sac.
And they stay fused for a long time, right? It's not until relatively late in gestation that they open.
That's right. They remain united until about postmenstrual week 24. This fusion period is critical for protecting the developing cornea and integrating the structures beneath. Separation is a process thought to involve complex tissue remodeling and keratinization, the maturation of the epithelial layer.
We need to distinguish between the muscles that move the lid and the muscles that move the globe, because their developmental origins are entirely different.
This is a classic example of the patchwork of head development. The orbicularis oculi, the muscle that closes the eyelids, develops from skeletal myoblasts originating from the second pharyngeal arch. This is ancient branchial arch material.
But the muscle that opens the eyelid.
A different story. The levator palpebrae superioris, the striated muscle that opens the upper eyelid, has a different lineage, derived from the neural crest mesenchyme. We see this mixing of origins everywhere in the craniofacial region.
Moving on to the lacrimal apparatus, the system for tear production and drainage.
The lacrimal gland itself, which contains the secreting alveoli and ducts, is derived from ectodermal epithelial buds that proliferate and invade the underlying mesenchyme. It's an ectodermal derivative.
And the drainage system, the lacrimal sac and the nasolacrimal duct, which handles the flow of tears down into the nose.
They derive from a solid cord of ectodermal cells located in the nasomaxillary groove of the developing face. This cord sinks into the underlying mesenchyme and then undergoes a complex sequential process called canalization hollowing during postmenstrual weeks 9-12 to form the finalized duct system. If this canalization fails to complete, newborns can experience tear duct blockage.
Finally, let's tackle the extraocular muscles. These muscles, which move the globe, the four recti, the two obliques, are arguably the most complex muscles in the body in terms of origin.
Their development is extraordinarily intricate. It requires the coordinated juxtaposition of specific muscle precursors, the outgrowth of specific cranial motor nerves, the third, fourth, and sixth, and neural crest mesenchyme contributions. The precursors originate very early in the precordial mesenchyme cells, which migrate laterally from the precordial plate region.
And these cells condense into premuscle cells in what the sources refer to as the head cavities, formerly the preotic somites. This is an ancestral segmentation of the head mesoderm.
Yes, a very specific, ancient developmental structure. These condensations later subdivide to form the specific muscles, each guided by its corresponding motor nerve. The sources highlight that myotube formation here starts later and progresses at a significantly slower pace than in limb muscles.
Why the difference in growth rate and structure compared to limb muscles?
It speaks to the specialization of the function. Extraocular muscles are not designed for bulk power, but for near-constant fine adjustment and fatigue resistance. Their structure reflects this. They are much smaller, the muscle fibers are loosely arranged, and they belong to very small motor units, allowing for incredibly fine, precise control.
And their molecular signature is also genetically distinct, which the sources highlight with the myosin isoforms.
Absolutely. Unlike limb muscles, which show a clear progression from one myosin heavy chain isoform to another, the extraocular muscles co-express two specific isoforms from the very earliest stages. This mixed expression pattern is unique and reflects their distinct contractile properties.
And to complete the picture of codependence, the muscle tissue itself is mesodermal in origin, but the connective tissue attachment, the tendons connecting these muscles to the sclera, is derived from neural crest mesenchyme.
That's a critical detail. You have a mesoderm-derived engine attached to a neural crest-derived cable, all controlled by a specialized cranial nerve. A defect in any of the three components can compromise eye movement, leading to conditions like strabismus.
So, by week 40, the architecture is largely complete, but it is far from functional mature. We need to discuss the unique characteristics of the eye at birth and the critical maturation steps that continue afterward. We must start with a major risk factor for preterm infants.
Yes, low birth weight and, crucially, high exposure to oxygen in incubators put preterm infants at high risk of developing retinopathy of prematurity, ROP. ROP is a proliferative retinopathy involving the excessive, abnormal growth of the retinal vasculature into the vitreous humor.
Let's look at the developmental logic of this risk. We noted that retinal vasculature only begins forming around week 15 and is still expanding peripherally at full term.
Exactly. The final stages of retinal vascular growth normally occur in a relatively low oxygen, high growth factor environment in utero. If an infant is born prematurely and placed in a high oxygen environment, that sudden change often causes the existing immature peripheral vessels to constrict and regress.
So the high oxygen basically shocks the system and shuts down vessel growth.
It does. Then, when the infant is taken off high oxygen, the now ischemic or oxygen-starved retina releases massive amounts of vascular growth factors in a desperate attempt to compensate. This results in disorganized pathological sprouting angiogenesis into the vitreous, which can lead to retinal detachment and blindness.
That makes the connection between developmental timing and postnatal environment incredibly clear. Beyond pathology, there are crucial developmental timelines for basic function. When do the critical reflexes that allow us to interact with light and focus appear?
The pupillary light reflex, or pupillary constriction, is entirely absent in fetuses and preterm infants younger than postmenstrual week 30. Variable responses only start developing around weeks 30 to 34. The neural circuitry for that reflex is simply not myelinated or integrated until late in the third trimester.
And what about focusing on things up close?
The convergence reflex, which allows the eyes to focus inward on a near object, typically appears only after postnatal month 4 in full-term neonates.
The sources also delve into the surprising visual environment in utero. Fetuses are generally not exposed to light, so what is the retina doing if it can't see anything?
What's truly fascinating is the discovery that retinal development in the fetus is highly active between postmenstrual weeks 22 and 40. This activity takes the form of intrinsic patterned electrical firing retinal waves that are essential for organizing the neural circuitry.
So the retina is practicing.
It's practicing. This activity is even associated with REM sleep, particularly between weeks 28-30. This suggests the retina requires constant, intrinsic, non-visual stimulation during this period for normal maturation and integration with the visual cortex.
So the brain is essentially learning how to process visual information using simulated input before the visual input even arrives.
Precisely. And the implication for preterm neonates is significant. By missing this critical period of endogenous stimulation, their retinal circuitry may fail to gain normal maturation, compounding the challenges already posed by ROP risk.
Let's discuss the physical differences at full term, week 40. The neonatal eye isn't just a miniature adult eye. Its physical configuration compensates for its size.
At birth, the eye is about 65% of its adult size, and it undergoes a rapid growth phase in the first year. Structurally, the globe is slightly more spherical than the adult eye, and crucially, the neonatal lens is more spherical than the adult lens.
And that's functional.
Highly functional. The increased lens sphericity increases its refractive power, which helps to compensate for the relative shortness of the neonatal eye. A shorter eye naturally focuses images behind the retina, so this powerful lens pulls the focus forward.
So the infant eye is beautifully engineered to compensate, but what about visual performance?
Neonatal acuity is quite poor, estimated to be about 2400. This is primarily due to the immaturity and sparse packing of photoreceptors in the fovea. This acuity improves dramatically as the cones migrate and pack more densely, reaching 2030 or even 2020 by 2-3 years of age. There is also a general trend for infants to be farsighted.
And finally, a great detail for anyone who has ever held a newborn tear production.
It's often a surprise, but even the complex lacrimal apparatus requires significant postnatal maturation. Preterm infants have reduced tear secretion, and the sources note that tears are typically not present with crying until more than 3 months of age in full-term neonates.
We have journeyed in immense detail from a few neural folds in the forebrain, governed by master regulators like PAX6-VEX wheats, all the way to the perfectly layered, self-sustaining visual apparatus, complete with its own fragile internal plumbing. It's impossible not to be impressed by the sheer number of things that have to happen with exquisite temporal and physical precision.
Our deep dive has illuminated several critical developmental themes. First, the incredible power of those initial transcription factors. PAX6-VEX establishing identity, and 7xx7 carving out space. Second, the crucial role of cell lineage and programmed cell death, evident in the conserved birth sequence of retinal cells and the necessary apoptosis of the optic fissure margins and the hyaloid artery.
And third, the dependence on cells from external sources, particularly the delicate balance required for the anterior segment, relying almost entirely on migrating neural crest cells to form the transparency of the cornea and the critical fluid dynamics of the aqueous humor drainage system.
And if we connect this back to the bigger picture, what stands out most clearly is the non-negotiable temporal precision required for vision to succeed. A molecular mistake early on can lead to cyclopia, while an environmental error in the incubator weeks later can lead to retinopathy of prematurity. Every single stage is a high-stakes engineering challenge.
And knowledge is most valuable when understood and applied. We saw one of those aha moments that directly intersects embryology and future medicine in our source material when discussing cell identity. We detailed how the retinal pigmented epithelium, the outer layer of the optic cup, retains the latent potential to become neural retina if the embryonic retina is wounded.
This raises an important question for you, the learner, to mull over and explore further. Considering the immense complexity of the adult layered retina, which has lost its regenerative capacity, and the fact that we know RPE retains this neural potential early on, what are the broader implications of such tissue plasticity when it comes to regenerative medicine? If we could effectively manipulate these ancient genetic switches, when ITF5 versus VSX2 and Donellers in an adult RPE cell, decades after the eye has completed its initial construction,
Could that offer a viable pathway for generating new functional retinal neurons to treat degenerative diseases such as age-related macular degeneration?